Hey guys, welcome to your third and final presentation for the DNA unit for Biology Credit Recovery. What I need you to do is find this worksheet within your credit recovery packet and take the notes as we move through the presentation. Make sure, as always, that if I move a little too fast, you pause the presentation in order to get all the notes you need. And those notes can then be used to answer the questions in the quiz at the end of this presentation. Now, in previous presentations, we talked about how DNA can make other DNA molecules. Now we're going to talk about how DNA can make other things, and they do this through a process called protein synthesis. Before we go any further, we need to go over a little bit of background information. First, we need to describe what amino acids are, and these are the building blocks of proteins. It's what protein is made up of. And then we need to talk about the two different types of nucleic acid used in protein synthesis. That's DNA and RNA. These are both words you should have seen before within this unit, so I'm not going to take too much time to go over what they mean. But there are a few new vocabulary words we need to talk about. The first one is codon. A codon is a sequence of 3 DNA or RNA nucleotides that correspond with a specific amino acid or stop signal during protein synthesis. Amino acids are the building blocks of proteins, and as before, it's pretty much what proteins are made of. And we also have RNA, and there are 3 different types of RNA. There is messenger RNA, or mRNA, and this is the molecule that carries copies of the instructions from the DNA to the ribosome, where proteins are made. Then we have transfer RNA, or tRNA, and this is the molecule that brings amino acids to the ribosome. And then, finally, ribosomal RNA, or rRNA. This is RNA that makes up the ribosome. Now, though DNA and RNA sound very similar, there's quite a few differences between these two molecules. DNA, for example, is double-stranded, the sugar it's made of is deoxyribose, and it has a different base. So DNA is ATCG. Now, if we look at RNA, RNA is single-stranded, its sugar is merely ribose, and instead of that T base, it has a U. That U then stands for uracil. So if we take a look at this picture, we see DNA on the left-hand side with its A, G, T, and C, and it has 2 strands, or 2 blue stripes, twisting. If we look on the right side of this picture, we have an RNA molecule, just 1 blue stripe, and it has C, G, A, and U. Now, every single time we make an RNA molecule, we'll be using U, or uracil, instead of T, thymine. Protein synthesis uses codons, which are composed of RNA, to code for one amino acid. Protein synthesis involves 2 steps, and those 2 steps are transcription, so that's the writing, and then translation, where we read what we write. First, let's talk about transcription. Transcription is the copying of the genetic message, so DNA, into a molecule of RNA, and this occurs in the nucleus. The steps for transcription are as follows. The DNA strand separates into 2 strands and serves as a template for the mRNA assembly. Free mRNA nucleotides match up with one of the single strands of DNA. The mRNA strand then leaves the DNA strand when it reaches a stop codon. And then the mRNA strand carries the instruction code out of the nucleus into the ribosome for protein synthesis. The second step of DNA protein synthesis is translation, and that's when the mRNA is used to assemble an amino acid sequence into protein. The steps of translation are as follows. The first step is when mRNA moves out of the nucleus and into the cytoplasm to the ribosome. Now, what I want you to notice is that the first step of translation is the last step of transcription, and that shows that translation comes directly after transcription. Then, once it's left the nucleus and is into the cytoplasm and to the ribosome, the mRNA is read by the ribosome and converted into a chain of amino acids with the help of tRNA. As the mRNA moves across the ribosome, the amino acids are joined together and they eventually produce a protein. The last thing we need to do within this unit is we need to learn how to use this chart. And what this chart does is it takes a 3-letter codon, like UCA, and it shows us what amino acid that's going to code for. The way we use it is if you look on the sides, we have first position, second position, and third position. And what that's going to do is it's going to tell us first position, U, and I'm going to circle that. Second position, C. I'm going to circle that column. Third position, A, which tells me that I'm making the amino acid, S-E-R. So that's the end of our final video presentation. There's one more video that you need to watch, and that just kind of does a detailed expression of DNA replication and protein synthesis, and kind of goes over the details. Please, if you are struggling with how to use the chart displayed in front of you, please come and talk to me. I am more than happy to help you. I'm here on Tuesdays from 3 to 5 o'clock and Thursdays from 3 to 4 o'clock. I'm also here for select Saturday schools. Please make sure to come get the help you need to be successful, and I look forward to seeing you in our next unit. Enjoy your quiz!